Biomicrofluidic Lab. The Biomicrofluidic Laboratory is located on the third floor of IEMN. Remove watch, ring, bracelet, and earrings. Careful hand washing. Tie long hair. Put a lab coat and an hygiene cap. Put overshoes. Wear gloves. Water bath. (3D bioprinter). Small CO2 incubators. Centrifuge. Syringe pump. Transmission microscope. Automated cell counter. Biological safety cabinet. Fluorescent microscope equipped for living cells real-time experiments. Cell subculturing techniques. Cell observation under the transmission microscope. Cell media removal. Cell trypsinization. Cell detachment. Cell media addition in order to stop trypsin action. Cell counting. Centrifuge the cells. Cell pellet observation. Cell culture supernatant removal. Cell media addition in order to resuspend the cells. Dispatching the cells in a 12-well plate. Cells observation. Cells are let inside a 37 degrees Celsius, 5% CO2 incubator to allow for cells adhesion and proliferation. Equipments to perfuse liquids inside microfluidic device. Negative pressure controller. Push-pull fluoagent. Adjust the negative pressure level to finally control the flow velocity inside the microfluidic circuit. Entrance, red ink-filled bottle. Flow sensor to monitor the flow inside the microfluidic circuit. Stopcock in case the flow needs to be stopped. Microfluidic chip inside the microfluidic circuit. Exit, waste bottle. Red ink flowing inside the microfluidic device. Control the flow velocity by controlling the pressure level. Syringe pump. Mid-pressure mise syringe pump. 2 syringes that feed 2 microfluidic circuits. Entrance, red ink-filled bottle. Microfluidic circuit. Stopcock in case the flow needs to be stopped. Biomimetic microfluidic device to stand for blood vessels. Nemesis user interface to control the flow. The flow level is set to 50 L per minute. Red ink flows inside the microfluidic device thanks to the flow imposed by the syringe pump. The microfluidic chip is observed inside a 800 m width channel. Inside a 400 m width channel. Inside a 200 m width channel. And inside a 100 m channel width. All channels are 100 m height. For biological applications, the microfluidic chip is put inside a 37 degrees Celsius, 5% CO2 incubator. 3D bioprinting. Select Bioprint. Select the 3D model you want to print. Select the surface on which you want to print your model between Petri dish, well plate and glass slide. Define the tool type. Define the bio-ink. Adjust the pressure of the pump in order to extrude ink from the cartridge. Adjust the printing speed. If a second tool is required for the printed model, repeat this procedure for the second tool. Preview the layers that are going to be printed layer by layer. Let's print. (3D) printed models of an ear and squares. Microfluidic and microscopy. Syringe pump Nemesis and Nemesis user interface to perfuse a liquid inside a microfluidic device. The flow inside the microfluidic circuit is set to 20 L per minute. Temperature controller unit to apply 37 degrees Celsius inside the microscope incubator box. CO2 controller unit to apply 5% CO2 inside the microscope incubator box. The microfluidic device is placed under the microscope, inside the microscope incubator box. LOSX application to control the microscopy experiments. Channels of the microfluidic device are observed under white light by transmission microscopy. Channel of 800 m width. Channel of 400 m width. Channel of 200 m width. And channel of 100 m width. All channels are 100 m height. Real-time microscopy experiment. Green fluorescent labeled cancer cells are injected in real time inside the microfluidic device, mimicking the blood vessels. Green fluorescent labeled cancer cells flowing inside the microfluidic channels of the chip. Observation of the interaction between cells that make up the wall of the blood vessels and the green fluorescent labeled cancer cells. In the first steps, cancer cells are rolling and then attaching to the wall of the blood vessels. Further fluorescent confocal microscopy experiments, at BICL platform, reveal that some cancer cells are able to cross the wall of the blood vessels. Understanding the extravasation mechanisms is important on the way to decipher cancer cells' dissemination throughout the entire body in the context of tumorigenesis. Photo patterning. Surface activation of a 6-well plate using a corona discharge. The surface activation can also be done using a plasma cleaner. A PDMS stencil is deposited inside the well of the 6-well plate. (10L of PLLGPEG at 0.1 mg per ml is deposited in each microwell of the PDMS stencil. The PEG is incubated for 1 hour. The PLLGPEG is rinsed 3 times with PBS before adding the photoagent PLPP. The 365 nanometers laser of the Primo system is turned on. After turning on the Leonardo software, the microwell of the PDMS stencil are defined. The desired patterns are launched. The number of patterns as well as the dose can be chosen by the user. Here is an example of circular patterns of 50, 100, 250 500 m of diameter. Primo photo patterning system allows to project on the substrate the pattern you want. The combined action of UV and the photoagent PLPP degrades the anti-fouling coating on the illuminated area. After the photo patterning experiment, the microwells are rinsed with PBS. An adhesion protein, such as fibrinogen or fibronectin, is added to promote cell adhesion. After adding the cell suspension, circular patterns containing cells are clearly observed. Only the circular patterns of 500, 250, 150 m are filled with cells. The outer region remains non-adhesive to cell.